Hello, good afternoon, everybody. My name is Hans-Dieter Volk. I'm clinical immunologist by training and was until last year head of the Department of Medical Immunology at the Charité and the BioH Center for Regenerative Therapies. And I was here in the function of the coordinator of the EU-funded project PACE. And we want to give you a sum up of the data we generated of this very interesting clinical trial and the accompanying science. Yeah, it's my pleasure to introduce this session, and I'm very happy to take over or give over the microphone to Norbert Weiss, who will give a general overview on the disease and the medical need.
Yeah, thank you. My name is Norbert Weiss. I'm a clinician. I'm a specialist in vascular medicine. I'm working at the University Hospital in Dresden. I'm head of the Center for Vascular Medicine in Dresden, and it's my pleasure to introduce you into the topic of the PACE project on critical limb ischemia. I have to share my screen now. Is it right? Okay.
Now it's coming.
Yeah. Okay, you should see the presentation now. I will talk to you briefly about the unmet needs in critical limb ischemia. Nowadays, the term is no longer critical limb ischemia. It's preferred to call it chronic limb threatening ischemia. And by this, explain you why we've chosen to set up the PACE study. I'm speaking on behalf of the steering committee for the clinical part of the PACE project, of the PACE trial. You can see the other members of the steering committee here. Chronic limb-threatening ischemia is a clinical syndrome that is defined as objectively documented atherosclerotic peripheral arterial disease in adults who must have at least one of the following clinical symptoms on sign. They must have either ischemic rest pain with confirmation of hemodynamically severe impaired perfusion of the foot. Clinically, that's usually manifested as blue ischemia, as shown here, or white ischemia, ischemic feet without lesions. Chronic limb-threatening ischemia may be manifested as diabetic foot ulcers or any other limb ulceration that is present for at least 2 weeks. Here you can see a limb ulceration in a patient that previously had been amputated, forefoot amputated due to diabetic foot syndrome, or it can be atherosclerotic peripheral arterial disease and gangrene involving any portion of the lower limb or the foot, as you can see in this clinical example. Peripheral arterial disease affects worldwide around 200 million patients. We've learned during the last decades that the prevalence is increasing, especially in low and middle income countries worldwide. It's a disease that is that prevalence is increasing with age, and the prevalence is about the same in men and in women. Of these 200 million patients with PAD, it's estimated that around 5 to 12% have the most severe form of peripheral arterial disease, chronic limb-threatening ischemia. That would be 20 million people worldwide. This most severe form of peripheral arterial disease, CLTI, is associated with a very high risk of both mortality and amputation. Epidemiological studies, recent epidemiological studies from Northern America or from the Scandinavian country give both mortality and amputation risk of 22% per year for patients diagnosed with this disease and who are not revascularized. The risk, as you can imagine, increases with the severity of CLTI and is much higher in patients who have wounds or gangrene. The only evidence-based treatment so far that reduces amputation and improves mortality in patients with CLTI is revascularization, is improving the markedly impaired perfusion. This can be done either surgically, by bypass surgery, thrombectomy, or by endovascular interventions with the aim at restoring flow into the foot by a direct line from the aorta to the foot. In clinical practice, however, revascularization is only possible in up to 70% of patients. I can show you data from health service research in Germany from patients that are treated in routine care. These are routine data from a big health insurance company of Germany, Barmer Ersatzkasse, that covers about 20% of the patients in Germany. Patients that are diagnosed with chronic limb ischemia in different Rutherford stages are revascularized in routine care in up to 70% of patients. Patients with wounds, however, are only revascularized in clinical practice in about 50% of the cases. These patients have in clinical routine care an amputation rate during 1 year of 21% and a mortality rate of up to 8%. The limits can be pushed. We have done a clinical registry in specialized vascular centers where we included 1,200 patients with chronic limb-threatening ischemia treated for and followed them for 2 years. And in this specialized vascular centers, the revascularization rate is not 70 to 50%, it's 90%. And the amputation rate can be brought down to around 3 to 4% over 2 years. So the limit can be pushed. However, not all patients can be treated in specialized vascular centers, and worldwide, definitely, these treatment options are not available. There are limitations for revascularizations. Mostly this is the anatomical unsuitability in patients that have long occlusions of the vessel, missing distal target vessels that can accept a bypass or a peripheral intervention or heavy calcification. I can show you this here, patients with a foot ulcer who has patent femoral-popliteal artery. That's the level of the knee, that's the thigh, and as you can see, the patient has no patent vessel in the calf going to the foot. So this patient won't have a chance of healing this foot ulcer and has no option for revascularization, as it's impossible to put a bypass just on very small collaterals of the foot. Other limitations for revascularization are comorbidities of patients, cardiopulmonary comorbidities, which do not allow long and complicated procedures, bleeding risk, or failed revascularization. The techniques for revascularization are rapidly or are developing, and we can push the borders even further. One example of these emerging techniques is transvenous arterialization of the foot in a patient compared to the case I just showed, which do not have patent vessels in the calf and no patent vessels in the foot. We nowadays can try to redirect the arterial blood into the veins of the calf with 2 catheters we put in the calf vein, and another catheter we put in the artery of the proximal calf. We make a connection between the artery and the vein on this level, stent the vein, and thereby redirect the blood into the vein of the foot, not the artery, which leads to retrograde perfusion of the capillary bed of the leg. That's possible even in patients who do not have patent calf vessels and may reduce amputation rate further. Technically, it's very challenging, and the first results indicate success rate of about 60%. If that's not possible, our treatment options are really limited. Of course, we can perform wound care. We have to do analgesia of the patients. We have to control cardiovascular risk factors. But besides that, there is no evidence-based treatment. Vasoactive prostaglandins, such as PGE1 or iloprost, have been used for a long time for the treatment of critical limb ischemia. Most recently, a larger clinical trial with PGE1 was published, which was randomized and placebo-controlled, included 800 patients and showed negative results. A Cochrane review of all published randomized controlled trials of prostaglandins compared to placebo in patients with critical limb ischemia showed that there is no difference between prostanoid treatment and placebo in regard to amputation-free survival, and amputation. So there is no evidence for vasoactive prostaglandins anymore. There is no scientific evidence for other methods such as spinal cord stimulation, sympathicolysis, or hyperbaric oxygen therapy. And there is no evidence for gene therapy aimed at transferring genes that promote angiogenesis in patients with chronic critical limb ischemia. The largest trial so far was the Tamaris trials, which tested gene transfer of the FGF gene compared to placebo in patients with critical limb ischemia. 250 patients in the placebo group, 260 patients in the gene therapy, FGF gene therapy group, and during follow-up of 12 months, there was no significant difference in amputation rate. Actually, there was a numerical trend in a higher amputation rate in the active treatment group. There was no difference in death rate. There were some more clinical trials that compared gene therapy versus no gene therapy, which are summarized in a Cochrane review or in 2018. And altogether, all these trials did not show a significant benefit of vasoactive gene therapy in regard to amputation-free survival, and also in regard to only amputation as the endpoint. There had been an attempt to use autologous cell therapy, bone marrow-derived mesenchymal cells for patients with chronic critical limb ischemia. Non-randomized clinical trials showed were in favor of cell therapy. However, after randomized, controlled, and placebo-controlled trials were performed, trials with low risk of bias, this effect of autologous cell therapy in patients with chronic critical limb ischemia disappeared. So we have no established and evidence-based treatment so far beyond revascularization. An alternative to to gene therapy or autologous cell therapy would be allogenic mesenchymal progenitor cell therapy with PLX-PAD cells. We are talking today. These cells, and you will later hear the exact classification of these cells. These cells are expected to have pro-angiogenic capacity, which might directly influence ischemia. They are expected to have antioxidative capacity, which might be important in treating critical limb ischemia because these patients often undergo ischemia reperfusion injuries that are associated with increased oxidative stress, and atherosclerosis is an pro-oxidant disorder. And as a third mechanism of action, these cells might have anti-inflammatory or immunomodulatory properties affecting this chronic inflammation that takes place in the chronic ischemic skeletal muscle. There are preclinical data that indicated efficiency of PLX-PAD cells in the classical mouse model of critical limb ischemia, the hind limb ligation model. Injection of the cells in a dose-dependent manner restored perfusion of the leg as measured by laser Doppler flowmetry. The effect was more pronounced when the cells were injected into the ischemic limb directly compared to the contralateral limb, but also worked in the contralateral limb. And histology also showed that these cells are possible to induce angiogenesis in the skeletal muscle. This mouse model, actually one has to say that's a major limitation of this mouse model. This mouse model is not a model of chronic critical limb ischemia, but a model of acute limb ischemia. The arteries are ligated, and immediately after ligation, the therapy is performed in otherwise healthy and non-atherosclerotic mice. Therefore, this data have to transfer into humans. There were 2 phase 1 open-label clinical trials with PLX-PAD1 cells in patients with chronic critical limb ischemia, 27 patients that received different doses of PLX-PAD cells, and either as a single or repeated doses via 30 to 50 intramuscular injections in the affected leg. This phase 1 trial showed that cell transplantation or the PLX-PAD cell injection into humans is safe without the necessity of performing HLA matching. The main side effects were injection site reaction, pain, muscle fasciculation, itching, or hematoma, and patient develop a bad breath as the cells are dissolved in DMSO. These phase 1 trials were not powered to demonstrate clinically efficacy, but showed that the amputation-free survival in these patients, which were no option patients, was 85 years. That is better than expected in this patient population. And there was a trend to improvement of surrogate markers such as pain and oxygen tension in the affected legs. As a follow-up, a phase 2 randomized double-blind placebo-controlled trial was performed to define the necessary doses and to confirm safety of this therapy. This time not in patients with critical limb ischemia, but with intermittent claudication. The trial was published at the American Heart Association meeting in 2018. And one of the major findings of this phase 2 trial was that the higher dose of cells, 300 million cells per injection compared to a lower dose, is more efficient, and is more efficient to use cells from 2 different donors during 2 injections 12 weeks apart compared to 2 injections from a single donor and only 1 injection. And this 2 injections with 300 million cells from 2 different donors led to a significant increase in maximum walking distance in these patients with intermittent claudication. Therefore, this treatment was chosen for the phase 2b/3 trial, the PACE-3 trial in patients with chronic limb-threatening ischemia. The main inclusion criteria of this trial were adult patients with age 45 to 99 with chronic limb-threatening ischemia caused by atherosclerosis. With minor tissue loss. That means all patients must have lesions, but must have a savable foot, defined by the Rutherford 5 category of chronic limb-threatening ischemia. The atherosclerotic peripheral arterial disease must be objectively confirmed, and patients must be unsuitable for revascularization, as I explained you before. These patients were then randomized in a 2-to-1 ratio, twice as many patients in the Verum arm compared to the placebo arm, to receive 2 injections of PLX-PAD cells in 8 weeks intervals into the affected leg. And patients were then followed up regarding the primary efficacy endpoint, which is defined as time to occurrence of major amputation or death, and also to a number of secondary and exploratory endpoints. And you will hear more details about the trial later in this meeting. So let me conclude. There is definitely unmet therapeutic need in patients with chronic limb-threatening ischemia that are unsuitable for revascularization, as we don't have conservative treatment options for these patients, and they have a high risk for amputation and mortality. PLX-PAD cells were promising. These are cells from young and donors that are potent, that are easily accessible in the required quantity. And preclinical and initial clinical evidence or initial clinical trials were promising regarding safety and efficacy. Therefore, PACE trial was performed in patients with ischemic foot lesions due to severe peripheral arterial disease. These patients received 2 injections of PLX-PAD cells from 2 different donors compared to placebo, and were followed up for 36 months. The clinical endpoint was a clinical relevant endpoint, amputation-free survival, which is definitely the most important clinical relevant endpoint in this patient, and we will hear the results later on during this meeting. Thank you for your attention, and if there are questions, I'm open for questions.
Do we have a discussion here?
We have a discussion.
Yeah. Is there any questions, comments here from the small auditorium? I think, no, thank you very much for this very nice talk, but I have one question to this very nice overview on the clinical trials which failed gene therapy and so on. You show only the odds ratio, of course, which is the most important one. But if you compare the placebo group in each of the arms, or in each of the different studies, are they similar or are completely different? Because we were very surprised from the relatively good results in the placebo group. And I would like to understand what was the placebo effect in the different trials. Is it comparable or from completely different levels?
Now that's a very important question, as over the time, over the years, we see that there is a trend to a lower event rate regarding amputation-free survival. This 22% one-year event rate that is published in recent epidemiological studies is even lower as event rates in the gene therapy, in previous gene therapy trials, and they are lower as in the PLX-PAD trials. So the natural trend in amputation-free survival is really an important question, and it's decreasing, probably due to better cardiovascular, general cardiovascular treating, because especially the mortality rate is decreasing to a higher extent than the amputation rate. And that's probably due to better systemic treatments such as statins and antihypertensives.
Okay, any other questions? Thank you, Norbert, for this answer. Okay, then we move forward to the next presentation. And it's my pleasure to announce Guido Moll, who is speaking on the work on the further specification of the cell product to better understand how homogeneous we are with a living drug and so on. Yeah, Guido, it's your stage, please.
Okay, my name is Guido Moll. I work as a system coordinator in the group of Hans-Dieter Volk, who is coordinating the PACE projects, work packages on behalf of the Charité group. I would like today give an overview on the in-depth specification of the cell product PLX-PAD. And we use the following panel of tools: genetic and epigenetic molecular profiling, flow and mass cytometry and cyclotron profiling, functional assays on immune modulation by PLX-PAD, and this, also a part of this work package was the establishment of the proof of concept of human organship model, and eventually to integrate all the molecular and functional data in a database to correlate with clinical outcome. As a short intro on the motivation for the work package 2 study, I would like to show shortly the slide from a blog post that I've seen like a couple of weeks ago from the last ICT meeting. And interestingly, the conclusion from the conference was that the heterogeneity and cell therapy products and processes and regular guidance is still a huge problem in 2022. And another very interesting point was also that many companies conducting these studies lack the required analytical capacity to conduct the adjunct analysis of their cell products in conjunction with the clinical trial. Indeed, this problem was already identified by the PACE consortium many years ago and was thus included in this project. So here you can see that the conventional characterization of MSCs is still insufficient nowadays. The first minimal criteria were proposed by Domenici et al. In Cytotherapy in 2006. And these MSCs...
Excuse me, Guido. Excuse me, I only can see the first slide, page 1.
Yes? Everyone else as well? Or
yeah,
can you...
We only can...
It's frozen.
Okay, it's still hanging on the first slide. Screen sharing is paused. Resume share. Can you see anything, or?
We can see, but we try to move your thing, and then you have to restart, probably. We can cut it later, no, Gabriela?
Try to unshare and then...
Dieter? Okay. Hello, my name is Guido Mohr. I work as assistant coordinator in Hans-Dieter Volk's group at Charité, who is coordinating the work package 2 on the in-depth specification of the cell product PLX-PAT. The tools that we use in this work package are the genetic and epigenetic molecular profiling, flow and mass cytometry and secretome profiling, functional assays on immune modulation by PLX-PAT, the establishment of proof of concept of human organship model, and eventually it will generate a database integrating the molecular and functional data. A short motivation for this work package in this study, I saw a blog post 2 weeks ago on the latest ISCT conference in San Francisco, USA, which gave some conclusions on opinion leaders of the ISCT committee. And it was concluded that heterogeneity in cell therapy products, process, and regular guidelines is still a big issue in 2022. Especially the industry lacks the required analytics, and this is still a hot topic in 2022. This problem was already identified by the PACE consortium many years ago and was thus included to accompany this clinical trial. Here you see the conventional, a summary that the conventional characterization of MSC like stroma stem cells is still insufficient. First definition of MSCs was given by Dominici et al. In Cytotherapy in 2006, and these MSC minimal consensus criteria are very useful but outdated nowadays. They give a poor differentiation of the cell product properties, considering macro and micro heterogeneity, for example, between different products such as PLX-PAT and PLX-RT produced by Pluristem, or cell batches, donors, and products manufacturing runs. Therefore, we aim to here characterize the macro and micro heterogeneity of the PLX-PAT product and other products used as controls. This serves to better identify distinct products and also to have a better idea on variation of donor to cell donor variations, for example, human donor variation or run-to-run variations in manufacturing. This could give a clear impact on the functionality and choice of clinical application, and also a rationale for designer cell products, for example, the anticipation of tissue factor expression in different cell products, considering their delivery. If these macro heterogeneity relevance, the size of this relevance is not clear yet, and it could, for example, used as for negative selection of poor runs or for correlation with clinical results. And here you see a short overview on some perspective articles that we have produced over the time of the PACE project since 2019, where we look at the intravascular MSC therapy product diversification and if it's time for new clinical guidelines, or also more recently considering MSC therapies for COVID-19, that we outlined in patients of patient coagulopathy, thromboprophylaxis, cell product quality, and mode of delivery for treatment safety and efficacy. And most recently in 2021-22, we gave an update on improved MSC minimal criteria to maximize patient safety. And we highlight here the importance of tissue factor and hemocompatibility assessment of MSC products. This you can see illustrated in these 4 little slides. So we have different types of MSCs, which are mainly defined by their source, such as bone marrow, perinatal tissue, and adipose tissue. And depending on the source, the cells can express different amounts of tissue factor. We only know that if we actually conduct in-depth profiling of the starting material to see this macro heterogeneity and this expression of this marker before we biobank the cells and want to use them in clinical trials. We can actually do preclinical testing with human blood, where we see how this tissue factor correlation, procoagulant tissue factor correlates with the clotting of the MSC when they introduce the blood. This will inform us on the necessary product delivery, the optimal delivery, its safety and efficacy, and co-treatments such as anticoagulation. And to highlight that the PLX PAT product is actually injected intramuscularly to avoid vascular administration. So here you see shortly the panel that has been employed in the PACE project. So we have the different starting materials, which have been characterized by various means, such as the KZ cell counter to analyze the phenotype and viability post-recovery of the cells for our lab work. Then we have employed next-generation sequencing, RNA-seq to analyze the transcriptome, and METHI-CAP to analyze the epigenetics and methylation status of the cell products. We have employed a flow cytometry, 262 surface marker CD screen, site of mass cytometry with a multidimensional analysis of 37 simultaneous surface markers, and also secretome analysis of 266 secreted analytes with the OLE platform, and various immune functional assays such as PLX endothelial cell immune interaction, immune modulation. And also we have tested human multi-organ chip models, and we have collected all these data and put them in a database for correlating the phenotypic markers with functional aspects of the product in vitro and in vivo. Here you see a first overview on the next generation sequencing method. On the left side, gene expression analysis of RNA-seq, and on the right side, epigenetic profiling with METHI-CAP. So we take the frozen cell product as provided by Pluristem. This is only for research grade, and then conduct mRNA isolation and Illumina sequencing, and then align the obtained sequences against the whole human genome to generate the sequence counts for all genes in the genome with the gene expression profiling. And in turn, for the epigenetic profiling, we use the METHI-CAP method, which is based on genomic DNA fragmentation, and then a capture of the CpG methylated double-strand DNA with specific dynabeats to then align this also against the human genome to look at the methylated and unmethylated regions in which gene clusters are transcribed or open. Here you see some very simple results from the NGS analysis. So on the left side, RNA-seq, in the middle, METHI-CAP. So you see that the PLX PAT cells form a very homogeneous cluster in the principal component analysis. So this analysis reduces the multidimensional data to 2 dimensions to better visualize them of the top 1,000 expressed genes. And we see that PLX PAT products are on whole transcriptome analysis extremely homogeneous. In turn, the other products, PLX Artene and PLX RAD, are slightly more heterogeneous, and but we also have included different development stages of the products. And also you can see that the PLX RAT and RAD product is closer to bone marrow cells than the PLX PAT product. And as a control, you see that umbilical cord derived MSCs, especially the ECFCs and UVEC cells, cluster very differently. METHI-CAP analysis shows that the PLX PAT product is slightly more heterogeneous on epigenetic level. So actually the transcriptome seems to be more robust than the methylome, and thus the methylome is more indicative of slight changes. But once more, we have a compact PLX PAT cluster compared to the other products. And here you see an overview on the surface marker screening with the CD screen. So this employs 242 specific antibodies directed against different CD antigens or standard antigens expressed on the surface of the cells, together with the isotype controls. And we have labeled different batches of PLX PAT and PLX Artene with this CD screen, and then obtained around 2,800 different FCS files, which were analyzed with bioinformatic analysis. And we found that, as you can see here in this heat map, which red stands for high expression and blue stands for low expression, that the PLX products are once more very heterogeneous and clearly distinguished from the PLX Artene products. Nonetheless, both major products fulfill the MSC consensus criteria. And we found that 60 of the 242 markers are expressed on PLX-PAT, of which around 1/3 of the 60 markers that are expressed are low or varying frequency. And as I said before, you have a very consistent positive, negative surface marker expression between different PLX-PAT donors and batches. So when you follow a line of a specific marker, you see that they are very similar. But we also have some characteristic differences between different product types. For example, the PLX18 cluster differently, and this is a combination out of differentially expressed markers. For example, PLX18 have less tissue factor expression, and interestingly, for intravenous application, they have medium expression of CD46, 55, and high expression of CD59, which makes them quite resistant to complement activation, as also seen for other types of placenta-derived MSC products. And then there's also some interesting donor-to-donor and batch-to-batch micro heterogeneity or variability in CD150, CD144, and CD128 expression, which is indicative of differences, potential differences in hematopoietic stem cell interaction, endothelial cell adhesion, or chemokine receptor interaction, which might potentially explain different capacity of the cells to respond to inflammation or modulate inflammation or migrate in vivo. Here you see an overview of the CyTOF mass cytometry method, which is the next method we employed after the CD screen for the initial screening. So based on the CD screen, we selected 37 promising markers. We included, of course, the ICT minimal criteria for good characterization, but also, for example, the hemocompatibility markers and different immunomodulation markers to characterize the product better. And the interesting thing about this method is actually that the cells are co-stained simultaneously with the 37 different antibodies, which allows to characterize them on single cell level. And here we have tested 7 PLX-PAT batches and 3 PLX-RAT R18 batches, again, at different stages of product development, and 1 bone marrow MSC line, and 3 replicates, which we used as a reference for bone marrow MSCs. And then we have loaded this on the mass cytometer, which then, based on time of flight, characterizes the single cells. And then we have obtained 15 SDS data files that were analyzed again with advanced bioinformatics, similar to the PCR analysis used in the next generation sequencing. We also can visualize multidimensional data in 2 dimensions via these t-SNE plots. You see t-SNE 1 and 2 dimension, and so on the left side, you see the cell product type and batch ID, and which shows you some major macro-micro heterogeneity between the different product types. PLX-PAT, PLX-Artene, and bone marrow MSC shown in red, PLX-PAT shown in green, and PLX-Artene shown in blue on the top graph. And then on the lower graph, considering the cell donors, you see that the 3 bone marrow MSC replicates all cluster together nicely in one cloud. Whereas with the PLX-PAT and R18 cells, there's some minor heterogeneity in the single cells. And when you look at the ICT criteria in the middle, you see that they're all consistently negative for CD45, which is a hematopoietic marker, a common leukocyte antigen, but they consistently express CD29, 73, 44, 90, and 105, although there's some minor variation in intensity between individual cells. And we also identified some interesting new targets for microheterogeneity, such as tissue factor, which is again very low expressed on the bone marrow MSC control, which is also found in other studies, whereas it is expressed at higher levels on the PLX PAT cells and R18 cells. And although we see that approximately half of the R18 cells are low in tissue factor expression, so there seems to be some heterogeneity there. And yes, so next, to not only look at cell markers or surface markers, we also looked at the secretome of the cells, since this is implied to have a major impact for their clinical suitability use. So we here use Olink technology, which is a proximity extension assay. You have this special probe that is produced by Olinks, and when it binds to an analyte with two antibodies, then it produces here a little oligo, which is then amplified PCR reaction to create unique double-strand DNA barcodes, which are representative of our targets. And these amplicons are then quantified by microfluidic real-time PCR to yield a normalized protein expression index. So it's like a delta index. And to choose what we want to analyze, we conducted a literature analysis of all previously known MSC factors, which were, for example, published within our Frontiers research topic with Martin Hocht and James Ankrum by a group from Portugal, including Ferreira et al., to look at who reviewed the MSC secretome. And they gave a very nice literature overview on many different factors and corresponding references and how they are expressed under hypoxia, inflammation, or 3D culture. So not just under static conditions. And based on this, and also the paper from Bluistam on human placenta-derived PLX PAT cells, which were stimulated with TNF-alpha and interferon gamma, we included actually the stimulation aspect to not just look at resting cells, but also at inflammation primed cells. And we then put together a panel of 260 analytes, which consist of inflammation and cardiovascular 1, 2, and 3 panel from Olink. So, and here you see the results. So at the top line, or let's start on the left, we identified 140 of the 260 analyzed factors are secreted in PLX PAT or PLX R18. And these include some hallmark mediators or regulators of migration, chemotaxis, growth, differentiation, proliferation, coagulation, angiogenesis. So lots of useful proteins secreted by MSCs that can have regulatory effect. And at the top line, you see the different types of products. So you see that the 10 PLX PAT products are extremely consistent considering their expression of markers, secretome, under stimulating conditions or under unstimulated conditions. And under stimulation, the secretome becomes more consistent than under resting conditions. So while the cells are resting, there's some micro variation, but when they're stimulated, they really boost their output. And similarly, the PLX R18 and rat product, although they are still in product development, from the batches that we had are very consistent. And they clearly differ from the bone marrow MSCs, which are somewhere in between, and also from the UVIC cells and endothelial cell, colony-forming cells, and umbilical cord cells, which are seen here in the middle. So this is very interesting results that we obtained here. So eventually we conducted many different functional assays, which were mainly done by Naima Soudi. And there's 4 main conclusions of these co-culture systems. First, MSCs, the PLX PAT cells have the ability to suppress allo-endothelial cell-induced T-cell proliferation, which varies to some degree between individual PLX PAT donors. So here you can see that there's some variation there between different products. Then PLX PAT induce regulatory M2, but not pro-inflammatory M1 macrophages. which you can see here on the expression of HLA-DR, CCR7, and CD206, which are characteristic for M2 macrophages. And then the third point is that PLX-PAT educated myeloid disease have a reduced potential to activate or stimulate T-cell proliferation. And eventually, PLX-PAT cells interfere with DC maturation. They use an incomplete maturation, reduce antigen-presenting cell capacity, and tolerogenic DCs. So altogether, the MSCs have an anti-inflammatory profile in co-culture systems in vitro, which is based on modulation of T cells directly, on macrophage polarization to anti-inflammatory phenotype, and also on modulation of dendritic cells, antigen-presenting cells. And eventually, we have put all the data that we have obtained in like the PACE database, which contains descriptive overview sheets on products and parameters, and calculation of average from primary data in the tabs, and you can see a basic design of the thing. And it's only useful if we can really correlate it with the clinical outcome. Unfortunately, in the clinical study, only 2 products were used and sequentially, so we cannot distinguish between the value of different products. As we see in vitro, some differences in immune modulation, if this translates in better or worse efficacy in vivo. But eventually, that was also a goal of the project, to use a consistent product with a reproducible phenotype in vivo. And here you see the wrap-up. First, next generation sequencing based molecular profiling with RNA-seq and MITIL-CUP. So some characteristic macro heterogeneity between different types of products, such as PAT or R18, and some micro heterogeneity within PLX-PAT products between different donors and batches, but also within R18. Second, the FACS surface marker CD screen and CyTOF single cell deep profiling confirms the macro heterogeneity of the products and micro heterogeneity of the donors and batches within a product category. And there's a robust expression of characteristic ICT MSC surface markers in PLX-PAT, but also on R18. And we found some new phenotypic and functional targets, such as tissue factor or programmed death ligand, which is important for immune modulation. And third, the Olink secretome analysis and immune modulation co-culture studies confirm that various mediators of angiogenic and immunomodulatory mechanism of action are produced by these cells. And there's a consistent response of PLX-PAT and R18 to TNF-alpha interferon gamma stimulation. And the cells have a very strong immunomodulatory function on T cells and myeloid cells in vitro. And I would also like to thank all the contributors to this work package on behalf of Hans-Dieter Volk, that team that he has built together with the other investigators of the PACE consortium to be able to conduct this analysis. Among others, Petra Reinke, Martina Seifert, and Dirk Strong, who also led some work packages, and also Uwe Marx from Tissues for providing this technology and sharing this technology with the group at BCRT. Thank you. This was everything.
Yeah, thank you, Guido, for the overview of the lot of data. Yeah, questions, comments from our team here, from our group. Of course, I think the good news are that we could confirm, even with deeper analysis, that the production manufacturing process is very robust, that you're getting almost similar cells. Of course, as a living drug, you can never get 100% identity. That is not feasible. But nevertheless, there are some interesting differences and between some micro heterogeneity, which of course is worth to follow up in the future. Particularly, if you use different lots. In the clinical trial here, we used only 2 lots for 2 different donors. But this was, of course, abolishing many of this micro heterogeneity problems or issues. I think it's not really a problem because we see a very minor differences. Yeah, any questions?
I have one question. Do you have any evidence or any hints whether or not the phenotype of the cells changes when they are in an organ? or in a mouse or in a human being? Do they keep their phenotypic properties?
Well,
that's difficult to answer.
The main question of my 5-year PhD and also subsequent postdoc was what actually happens with the cells after you give them in vivo.
Yes.
Mostly asking after intravenous injection where they disappear very rapidly.
Yeah.
Tissue injection is a whole different story. The cells persist much longer and seem to integrate into the tissue. So if you do actually look into the tissue, the cells stay there for, instead of like a couple days, up to weeks, depending on the model system used. When you use an animal system, in humans, I don't know how long they persist in vivo, if that has ever been analyzed. In blood, we have done it after infusion of patients. We have looked and we can see them for 1 or 2 hours and then they disappear from the circulation and are all in the lungs. And one big issue is the freezing thawing, of course, of the cells. If you inject a frozen versus a fresh product, how the cells integrate into the tissue and survive in the tissue, if that has an influence. And but nonetheless, lots of analysis has been done on that, showing that even frozen product can yield a good response when infused or injected into the tissue. So alone to give the cells, either fresh or frozen, seems to be sufficient to actually induce some good response in vivo. And that's very, very interesting to actually decipher what actually the mechanism of action is, how the cells respond to the body and how the body responds to the cells, and what's actually the mechanism of action there. If it's really the secretion of factors, or if it's a contact-dependent interaction, if it, many people are now on the train to look at education of the macrophages and antigen-presenting cells, how this changes upon MSC application and yields to a transient immunomodulation, which in turn is favorable for tissue regeneration and angiogenesis. And another aspect is also the recruitment of endogenous repair cells, such as endothelial progenitor cells and other cells that mitigate or mediate tissue repair. So there's is a very interesting crosstalk, and it's not so clear what one can learn from animal models. I think it's more necessary to look at biomarkers, what Dieter will present, to actually know what's happening in the human patient, because there's a lot of cross-oncino reactivity in animals, and animals are not really representative of humans when it comes to mechanism of action in many ways. So you can get some interesting hints, as you indicated with the CLI model, the acute versus the chronic model. But in the end, if you have a patient which has been suffering for years under tissue decline and lack of oxygenation, that's probably a whole different story compared to a mouse.
Yeah.
Yeah, I think maybe some comments to this. I think it's clear that it's a plus and a minus of this of a living drug. On one hand, you have a lot of adaptation to the environment. That is a good thing, because therefore the cells much better than a single factor you inject. And second, of course, it's making for a pharmacologist is it a nightmare, because you cannot predict all biological effects. Precisely regarding to the PX cells, we know 2 things. First, we know from different experiments at Lewiston in our lab, if you put it in a different environment in vitro, you have a lot of adaptation. For example, in the inflammatory environment, hypoxic environment, you're getting a different profile regarding secretome, gene expression. Interestingly, not a methylation. That means the cell is stable. We cannot change the methylation pattern, only transcriptional level and proteomics level, showing that the cell by itself is relatively stable, but function which is called is different. A second argument comes from the experiment with mice. If you inject the human PX cells in the mice, you see, of course, you can measure the cytokine response, the human cytokine response as measuring of the direct effect of the cells. And you see, if you treat the mouse with, for example, with radiation or other stress factors, you see a difference in the higher cytokine profile than with in a healthy mouse, indicating that the cells sense in vivo danger signals by ischemia, by tissue injury, and so on, and are more active. But in the mouse, I have to say, we have not seen a completely different profile. it's more quantitative than and how long it expressed than a qualitative difference. Okay, if you have no further questions, we move forward to the next one, given by Nitzan on the PACE clinical study, the core of this study. Thank you, Nitzan, for giving this talk.
Thank you, Dieter. 1 second, I will share my screen. I hope you can all see the slides.
Yes.
Perfect. So thank you for this interesting introductions and talks that were provided to really set the stage for this presentation of the clinical study results and some of the challenges that we had along the way. My name is Nitzan Halevi, and I'm the Chief Medical Officer for Pluristem. And I want to share with you today the results of the phase 3 PACE clinical study, which was a randomized, double-blind, placebo-controlled, multi-center study run in parallel groups as described by Norbert in the first talk, aiming to evaluate the efficacy, tolerability, and safety of IM injections of PLX-PAD in no-option patients with CTI. So just as a quick reminder, as this was detailed in the first talk, the study was run in 54 sites globally. Actually, only 42 sites ended up recruiting patients, and it included patients with Rutherford stage 5. So they all had to have minor tissue loss and be no option patients. So they had to be unsuitable for revascularization to be able to qualify for the study. 213 patients were then randomized into either active PLX-PAD, 2 injections at day 0 and 60, or placebo in a 2-to-1 ratio favoring active treatment. The primary endpoint was the time in days from randomization to occurrence of major amputation of the index leg or death, or otherwise amputation-free survival. And patients were to be followed up for at least 12 months, but as long as 36 months of follow-up based on the study design. Our first patient was recruited into the study in August of 2017, and the last patient had their last visit in February 2021. So quite a long recruitment period with some challenges along the way. The study was stratified by 4 major criteria, which I'd like to explain, as these are important when we analyze the data to understand. The first was the severity of the wound and the wound area. We tried to include both of these parameters into one score, where we defined patients with a low severity wound area as patients that had an ischemic lesion smaller than 10 centimeters squared and no black toe, or a single black toe with no lesion. And then patients with a high severity as those that had either more than 1 black toe or a lesion that was 10 centimeters or larger. Patients were also stratified based on their diabetes mellitus status at study entry, if they were diabetic, yes or no. Their region, obviously, we know there's a lot of heterogeneity in care. So it was very important for us to stratify between lesions in this multinational study. And then the number of lesions, at study entry, 2 or less or more than 2. So as I said, and I hope you can see my cursor, we randomized 213 patients into this study, 143 to the PLX-PAD arm and 70 into the placebo arm. We ended up treating 203 patients, 134 in the PLX-PAD arm and 69 in the placebo group. About 70% of patients completed 12 months, and a very small amount of patients completed the 36 months visit, mostly because the study was terminated early. The most common reason, as I said, for early discontinuation was the termination of the study. We also had a number of patients who passed away during follow-up. From demographics and baseline characteristics, the majority of our patients were male, 76%, mostly Caucasian, and then mostly non-Hispanic or Latino. The mean age was 70 years. and the mean BMI was 27. Most of our subjects were ambulatory when they entered the study, 86%. The mean area of the ischemic lesions was similar between the two treatment groups at about 5 centimeters squared, and the median was around 2. You see here some patients were really were outliers as to the size of their wound, really driving up the mean versus the median here. As to the number of lesions, the mean and median were fairly similar, with about 2 centimeters squared. Sorry, 2 lesions, mean number of lesions per patient. Most of our patients were recruited in Central and Eastern Europe, with a minority in Western Europe and North America. And then the majority of our patients were defined as low risk based on the criteria that I shared with you earlier, either a lesion that is smaller than 10 centimeters squared or 1 black toe. 60% of our population was diabetic at baseline, and most of the patients had 2 lesions or less, 80%. As I said, our analysis for the primary endpoint was a time to event analysis. And just to explain, patients were contributed time on study until they were censored, either at the end of the study or if they were terminated early, the time of their early termination. They contributed time on study until they had an amputation. We continued follow-up, obviously, after the amputation, but that event, the amputation event, was their contributing event. Similarly, if a patient underwent an amputation and later passed away, the time that was measured was only the time up to the first event of amputation or deaths. So as I said, our primary endpoint was to be measured at 36 months, including all patients and all time points in the study. This is for the intent to treat population, as was planned in our statistical analysis plan, including 213 patients. And we're looking at time to major amputation of the index leg or death. First, I want to look at the number of events of either amputation or death. We had 53 events overall, 29% in the placebo group, and 23% of patients in the PLX-PAD group experienced an event. That translated into an event rate of 0.9 hazard ratio, which obviously was not statistically significant. So the study failed to reach the primary endpoint. This was already very evident in an interim analysis that was conducted, and therefore the study was terminated early based on the low chance of it hitting the primary endpoint. If we look at a Kaplan-Meier survival curve of amputation-free survival, you see here that really the differentiation in curves for PLX-PAD in blue versus placebo in red starts after the second injection following the 60-day visit. And the curves actually stay separated for the duration of follow-up as long as 36 months. Obviously, this separation in the curves is not meaningful enough. We also looked at the modified intent to treat, so 230 patients who were actually treated with their assigned study drug, either placebo or PLX-PAD. And while this slightly improves the hazard ratio, of course, this is still far from being statistically significant. An additional sensitivity analysis is to look at the individual components to see if there's a differential impact on mortality at the top or major amputation in the bottom. And you see really that there's no statistically significant or clinically meaningful difference, with about 11–12% of patients having or dying on study for both treatment groups, and about 16–17% amputation in both treatment groups. The finding that was actually quite anticipated is that most of the events in our study occurred within the first 12 months. It's important to mention here that the actual placebo event rate for the first year was 24%, while the study was really sized for 35% of patients experiencing either major amputation or death within the first 12 months. But since most of the events, 80% of the events, or 42 of the 53 events occurred in the first year, we wanted to look in a post-hoc manner in only those events that occurred until the first 12 months. And here you see some separation with 24% of patients in the placebo arm versus 17 or 18% of patients in the PLX-PAD arm experiencing an event in the first year. Again, translating to a hazard ratio of 0.8, which is encouraging, but still very far from being statistically significant. For safety, obviously, this is a very sick population, as was described in the initial talk by Professor Weiss. And most of them had at least one treatment emergent adverse event. Okay, so about 80%, 70% in the two different groups had at least one treatment emergent adverse event. Only 60% of placebo patients and about 50% of PLX-PAD patients had any serious treatment emergent adverse event. And most of the events were not considered related to our study drug. The most common events that we saw in the patient population were very similar between the placebo and PLX-PAD groups, and very common in this patient population, even without any treatment. And we're talking about ulcers, gangrene, extremity necrosis, ischemic limb pain, and peripheral ischemia. So no really surprising finding as to the very common adverse events that were observed in this patient population. It's what we would expect from this patient population. Importantly, of course, as mentioned, this treatment is being injected intramuscularly. And so we took a very close look into the incidence of injection site reactions, which we anticipated to be more common with active versus placebo. And indeed, they were slightly more common. Injection site pain occurred in 7.4% of patients with PLX-PAD versus 5.8%. We also had a handful of patients experiencing muscle spasms and involuntary contractions. All of these were self-limiting and none were considered serious. As to major adverse cardiac events, these events are obviously very common in this very vascular patient population. And indeed, we saw several of those in the study, and I'll go through the overview now. For myocardial infarction, we had 5 events overall, 3 on PLX-PAD and 2 on placebo. I remind you that we dosed twice as many patients on PLX-PAD versus placebo. All of the events were assessed as non-related to study treatment by the investigator, and all in patients with multiple cardiovascular risk factors. We had 5 events of stroke. All of those were in the PLX-PAD arm, all assessed as non-related to study treatment, and all in patients with multiple risk factors or previous stroke prior to study entry. Importantly, and since all these events occurred on PLX-PAD patients, we went and searched for the incidence rate in the literature to compare it to our incidence rate. The incidence rate that was observed in our study is 2.4 per 100 patient years, which is similar to what we see in a recent publication, 2.5 per 100 patient years in CLI patients. So within what we would expect in our patient population again. For cardiovascular death, we had 5 patients passing away due to a cardiovascular cause, 4 on PLX-PAD and 1 on placebo. Again, I remind you that we dosed twice as many patients on PLX-PAD versus placebo. And then 3 subjects passing away from sudden death, which is it's very common to include in this category since we didn't know the cause of death. 2 were on PLX-PAD. One of those was one of the stroke patients, and then one on placebo. This is just an overview of all treatment emergent adverse events that concluded with a fatal outcome. And you see here that there's really no clustering into a certain cause of death that would cause us any concern or alarm, but patients obviously with long follow-up and very sick patients die of multiple reasons on study for both treatment arms. As to malignancy, this is always something that we keep in the back of our minds when we're treating with MSC, very potent cells. And we see no imbalance here in malignancies here, with overall 4 malignancies reported during the study, 3 in the placebo arm, and 1 for PLX-PAD. And again, no clustering of a specific malignancy, so no concern observed here. So before I conclude, I wanted to talk a little bit about the key challenges with the study. As I showed you, the placebo event rate, which was much lower than expected, especially in the first year when we expected most of the events. And the study was sized based on a 35% event rate for amputation and death in the first year. We only observed 24% of patients experiencing an event. And this goes back to what Professor Weiss presented and talked about, that yes, probably the sizing of the study was based on previous data where the quality of care for these patients and control of their cardiovascular risk factors really assists in reducing the overall event rate, which is fantastic news for the patients, not so much for our study. We had multiple challenges with recruitment, with non-recruiting sites and a very slow recruitment rate. This did not improve with COVID-19 hitting in early 2020 as well. So we had the challenges of conducting a clinical study throughout the pandemic. We also had multiple missing assessments, mostly during COVID, but also before. Peripheral pulses and other assessments were missing, which really hindered our ability to analyze the data appropriately. Obviously, for our primary endpoint, that is a very robust clinical endpoint. That was not a great issue, but for some of our secondary endpoints, this was more problematic. So to conclude, as I mentioned, this study was ended early for futility when a predefined criterion was met, and it was clear that the study would not reach the primary endpoint. We do note a separation of the amputation-free survival curves at day 60 following the second PLX-PAD administration. We did observe that, as expected, most of our events occurred within the first year of treatment. And PLX-PAD was found to have a favorable safety and tolerability profile for this target population. While we observed stroke cases on PLX-PAD treatments, treated patients only, the observed incidence was similar to the reported rates in the literature. However, as an abundance of caution, we will evaluate stroke as a potential signal in future studies with PLX-PAD. And with that, I'd like to conclude and thank you for the invitation to speak today.
Thank you so much, Nissan, for this overview on the clinical results. How would you, as the question goes to you, but also to Norbert, what would you say, what is the conclusion for the future? What can we do with this result? Because that, of course, on one hand, we have a lot of information, and on the other hand, of course, overall results is disappointing, we have to say. The question is, how do we see the future? As a question goes to both of you, and maybe also to the round.
Norbert, I'll let you go first.
Mm-hmm.
Okay. There are several issues. First, I think we have learned a lot about natural history of CLTI nowadays. That's very important, and especially we have learned that amputation rate and mortality further diverse, and we can influence mortality, but we are worse with influencing amputation rate. Second, I think we should more closely look into subgroups further, looking at comparing diabetic and non-diabetic patients. Maybe you can comment on that because you have already done the subgroups. Third, the curves separated, started to separate after the second injection, and all events occur, or most of the events occurred during the first year. So the question is whether or not it's necessary to give boosters, maybe a third or a fourth injection during this very critical first year of these patients. These are that probably would be the most important topics for me.
Mm-hmm. Thanks, Norbert. Something additional, Nissan?
Yeah, very, very valuable comments, and I thank you. I'm sure all the detailed results of the study will be the topic of a future publication. As to additional dosing, that's something that we obviously considered following, as you said, seeing the separation in curves. We need to keep in mind, of course, the balance with repeated injection of allogeneic cells and what that may potentially, do from an immunogenicity perspective. But that is something that we will continue to evaluate, definitely.
Other questions, comments? Okay, then we go over to the biomarker part. I will present. I'll try to share. Can you see it?
Yes, but not in screen share mode. Not in presentation mode.
Can you see it now?
I can see it.
Is it in the right way, or do we see still the
switch in presenter mode? It's not in presenter mode. You can just need to swap.
No?
Perfect.
Perfect. Good. Thank you so much. Okay, I like to summarize the work package on the biomarkers for therapy monitoring. As mentioned before, we wanted to learn as much as possible from the clinical trial by mechanistic site studies. On one hand, a deeper analysis of the cell product, and on the other hand, a deeper analysis of the patients how they react to normal placebo treatment as well as to the therapy. I think that is work from many partners, of course, from the clinical study partners, from Pluristem, from the sponsor, from ICON, and many others, also from different facilities, from our team, in particular the study lab and the bioinformaticians, and the HIA lab, Nils Nachmann and Guido helping and also project coordinating. I think the specific task on biomarkers was clear. We focused to blood biomarkers. Unfortunately, we did not get the permission to make biopsies, and that means we have to look for the indirect effects that we can see in the circulation. And we wanted to address safety issues, PD as a pharmacodynamics, mode of action, and therapy response stratifiers. And we use different qualified or even validated test platforms, simple ELISA tests, multi-parameter flow cytometry with a CE-labeled machine and a fully validated test portfolio, multiplex ligand assay based on the Mesoscale platform. You can make in parallel measure different parameters, for example, cytokines, CRP, and so on. And molecular analyzers RNA-seq from whole blood sequencing. I think the principal flow was, of course, the biosample availability, clinical site partners, patient screen recruitment, study visit compliance, time and quality. Sounds easy, but it was a very big task because of the increasing number of clinical sites because of some non-performing or non-recruiting sites. Therefore, we had to go to many other centers to organize the logistics, the quality, the training of the staff. Therefore, we have not as many samples we wanted to have. Transport, shipping, then the fresh analyzer, for example, some markers like the NOT cell markers, you have to measure immediately. That we can do it only from centers with short transport. And others is easier if you can make a cold storage or even a deep freezing storage. And finally, of course, all the data were put in the certified laboratory information medical system, the LIMS system, which is GMP compliant and can be later also linked with the database from study data and patient cohort data. And I will now show you some, I picked only some aspects because there's too many data and not all are ready. The inflammation related parameters, the first one. And here we wanted to address three hypotheses. One regarding safety. We postulated there's no cytokine release after PX cell therapy, despite the use of unmatched allogenic cells, and even more, despite repetitive injections. For example, if you have an allosensitization, you can expect after the second injection a stronger cytokine release. Second, the pharmacodynamics or the mode of action analyzers regarding, we speculated that inflammatory parameters might be increased in patient plasma from such kind of patients and will trend to normalize in response to PX-PAD therapy. And third, we hope that we can find inflammatory parameters which predict the outcome regarding response or non-response. And here we use cytokine CRP measurements in plasma by multiplex ligand assays, which are very sensitive, have a high linear area of measurement, and very, very robust and highly validated. Here show you one example. You see that many parameters, you see gamma interferon, IL-10, IL-12, IL-13, IL-1 beta, IL-2, IL-4, IL-6, IL-8, TNF-alpha, and CRP were measured in a multiplex format. And you see that many cytokines you could not detect, or very, very few patients only at all. But others, which you can see here in red and green, you can see, you can measure, you can find significant levels. And red is just before injection, and in turquoise is 4 hours after injection. You see day 0 in the placebo and month 2 in the placebo group, and the same for the PX group, day 0 and month 2. Month 2 is immediately and after the second injection to see is there any effect on the second cell application. Overall, you can see, without going through the details, there is no significant cytokine release post-PX cell therapy, despite use of unmatched allogenic cells and repetitive injection, meaning our hypothesis was fully confirmed. Regarding the second hypothesis, can we see a positive shift of inflammatory parameters in patient plasma, because we know that PX cells are able, at least in vitro, to act anti-inflammatory. But here we can say there's not so much effect. There's some individual patients, but overall, there's no dramatic change in the cytokine levels before and after treatment, both in the placebo and the patient group, the treatment group. The third question was, is there any evidence for therapy response stratifier? Can we see that inflammatory parameters might predict outcome or might early show evidence for a positive or negative outcome regarding response? And interestingly, what is here shown that you see for IL-6 and even more for CRP, higher levels in both in the placebo and in the PX-PAD group before treatment as a baseline, the upper top, in such patients who developed event as a death or amputation in the first year, the turquoise versus red one. You see the significant higher levels, but again, it's not for all patients if you see the individual points, but nevertheless, it's a clear, even significant difference between the baseline levels in the responder or non-responder, or other words, independent from placebo or other persons, indicating that hyperinflammation obviously is hardly to overcome even by cell therapy. And next, if you look after 2 months in the samples, you see interestingly that patients with events who develop events within the first year, as the Turkish one, express lower interferon gamma levels and higher IL-6 levels, but interestingly only in the PX PAT group and not in the placebo group. If you look for the levels and compare it with the pre-treatment levels, then you see it's not an increase, it's a no decrease. That means, in other words, in the PX treated patients who are not responding, that means they develop events, there's no decrease in IL-6 in contrast to the other groups where you see it. The question is non-control of inflammation a negative predictor? And is there any relation to other co-founders, for example, type 2 diabetes, which was shown an independent study from us that we see more inflammation in type 2 and decompensated type 2 diabetes patients. That is, of course, worth to check. Next, we look for NOT cell related parameters, as mentioned before in Guido's talk. It's absolutely clear that PX cells have a strong impact in vitro and also in animal models on the functionality and survival of endothelial cells. And because we could not take biopsies, we asked, is it possible to analyze the circulating endothelial cells? It's a very tiny population in the circulation, only a couple of cells per million total cell counts. But we have developed in the past a very highly validated and robust method by multiparameter flow cytometry to detect both the mature circulating endothelial cells, so-called CECs, as well as endothelial progenitor cells, so-called EPCs. And the question was, are the abnormal frequencies we have seen in the previous study in CLTI patients will trend to normalize in response to PLX therapy? And can this response be associated with clinical outcome? And the data show here, the problem is here we could do it only with a couple of centers because it needs fresh transport, and because it's sensitive, we cannot transport it too far. And we started relatively late to have really a very robust assay. And in the previous study from Maura Farnacci in our lab, we could show that CLI patients have higher levels of circulating endothelial cells, CEC, compared to age-matched and sex-matched controls. And what we could see in our study, in the PACE study here, that our baseline levels in our PACE studies are even higher than in the previous CLI. That means it's really severe CLTI patients with high levels of circulating endothelial cells as a typical marker for endothelial cell injury, and then the leaving the vascular system and circulating in the peripheral blood. And you see in the placebo group is almost no change compared to the baseline after 6 months. But interestingly, in the PX group, we have a clear trend down to normalization by more than a factor of 4, going the number of and coming in the direction of normal cells. And in some patients, we even completely normalized or even very low levels of circulating endothelial cells. Similar for the EPC, even here we see higher levels than expected from the previous study in the baseline. But we see here also the trend, particularly if you show the low expressing patients, compared to this group, we have no longer a low expressing patient. There's a similar trend. If you mix a ratio, it's highly significant difference. Of course, it's now interesting to see is this response of CEC, EPC associated with clinical outcome, because you see there's individual differences, and that is not finally analyzed. That will become in the next couple of weeks. Next topic is, or next tool we use is a gene expression profiling using the RNA-seq technology in whole blood by next generation sequencing. we used whole blood. There's no any manipulation. We have, that means we have a reflection not only of the gene expression per cell, but also of the cell composition, because of course there's different profile in different cell subsets. And the hypothesis was gene expression profile will trend to normalize in response to PX bud cells. The problem is here that we have not enough, we could not get sufficient numbers of sample size, so that we can only make trend analysis. And we have also in about 50% of samples a methodical failure. It did not work. It did not pass the quality criteria. That is not a problem of the lab, because in a parallel study, in the HipGene study, is also using PX cells. Almost all samples are working. That means it's obviously a transport issue or a collection issue in this study. Nevertheless, the samples we could analyze, we can say there's a similar clustering of patients. That means if you have 2 red dots here, for example, or 2 blue dots, meaning that the patients before and after treatment are similar clustering, but individually completely different. That means we have a stronger difference between patient to patient than patient before and after therapy, indicating that the sensitivity of the method is not high enough to see in the peripheral blood changes induced by the PX treatment. Last, the alloreactivity. Of course, it is a safety issue, as we speculated or hypothesized that as in previous phase 1-2 studies seen, we do not expect any negative impact of alloreactivity on clinical course regarding adverse effects and therapeutic efficacy. We use 2 tools, T-cell response on a few patients, because it's the same problem, because you can only do it from properly prepared and transported samples, and alloantibody response from serum, which could be done by almost all patient samples. And what you can see is that the analysis is not completely finalized. What we see, we could, from 213 patients, which were recruited from 200, we got from 3 visits material for the HLA lab, which is quite nice, that we can make really a clear answer regarding the alloreactivity. And simply, we can categorize the types of response in different categories. If some patients have before treatment, no any allopresentation, and afterwards also not. That is a majority of patients, confirming the previous data that most patients do not show immune response to the allo. Then we have a category of patients who have allopresentation before against multiple HLA molecules. We have also seen before, particularly in the elderly population. That means they are, and after treatments, they are still allosensitized. And here we have 2 outcomes or 2 causes. In some, the titer and profile is stable. In others, we see quantitative, meaning the titers go up, or the mean fluorescence intensity goes up, or even down. We also some down. Obviously, qualitative changes regarding the number of HLA molecules recognized by the panel-reactive antibody. That is a typical profile we see also after allotransplantation. But that's only some patients. And also interesting is that the majority is, we are speaking, no, that's almost all. Then we have the third category, no allopresentation before, but allosensitization afterwards. That looks like there is a specific immune response against the PX cells. And again, we have here 2 types. One is a polyclonal response, meaning we have many specificities against different antibodies. We have seen it also in the past, and here we see it in some patients, indicating there is a polyclonal response of B cells or pre-activated B cells to cytokines like IL-6 and so on. But we have also some patients with PX-specific, so-called donor-reactive antibodies. There are 2 or 3 are still in the final analysis of the 200 patients. That means it's a minority, but say 2 or 3 patients have significant titers and also persistent. And that is also an important point. The polyclonal response in many patients, it goes down after a couple of months. So it means, in summary, we see here shows the quantity. You see that here, these 3 patients in particular are very high, higher than normal, which show a clear HLA class 1 reactivity, which are related to a donor mismatch between the PX cells and the recipient. And that could be, of course, could be de novo antibodies, but it can also be upregulated response to other antibodies as a polyclonal response that we are checking at the moment in detail. Almost nothing happens with the class 2 antibodies. There's some noise, some very weak expression, and some positivity, but you see it if you compare it with the placebo group, is almost practically no patients who go upper the threshold. That means, in summary, we could say that our hypothesis is more or less confirmed. We don't see any clinical, visible clinical adverse effects. We see some alloactivity, but not too much. But in some it's stronger, as mentioned. And the analysis regarding the impact on the therapeutic efficacy is ongoing. Last but not least, we tried also to develop new biomarkers assay, which could not be ready for this ongoing trial, but for the future. And for example, as the endothelial cells are important, we wanted to learn more from the endothelial cells than just counting the cells in the circulation, and wanted to make a marker profile which could be related to the PX cells. And this was done in our lab to co-culture PX cells with endothelial cells and without monocytes, with and without inflammation, and then making RNA-seq analysis and see different regulations in the genes regarding extracellular matrix organization, blood vessel development, angiogenesis, and so on, and so on. And most importantly, PX cells induce a specific pattern in endothelial cells, and even more if monocytes are present. You see it here, for example, in the right hand, the upregulation versus the controls, and even more if you have monocytes in addition to culture. And that such kind of markers, for example, platelet-derived growth factor receptor B or PGF and so on, that could be markers which can be used on the circulating endothelial cells to monitor for the future better the functionality, not only the number of endothelial cells in the circulation. Okay, in conclusion, despite some logistic problems in sample collection and transport, the study revealed quite interesting results. High inflammation at baseline looks like it's a hint for poor response. It might be related to type 2 diabetes. There's no relevant safety issues regarding repetitive off-the-shelf PX application in direction of inflammation and allo response, at least if it's used in the way here in the study, in the distance of a couple of weeks and different products. Despite formation of some alloantibody formation, which looking at polyclonal than specific and only very few examples as mentioned before. Reshape of altered circulating endothelial cell EPC ratio by PX therapy, confirming that the EC function is a target of PX cells, also in vivo, not only in vitro. What does it mean for the effect? It's not clear because you see it obviously also in overall, because we don't see a clinical effect of the therapy, but we see only with PX cells the effect on the EPCs. Then the question, is it an epiphenomenon, or is this alone not sufficient to help for the better functionality? Need for further analysis is of interesting biomarker change in relation to therapy response is ongoing, particularly for the CSC EPC, the inflammation, and the alloantibody response. Last but not least, I'd like to thank again all the people I mentioned before on the first slide who contributed to this trial. Yeah, thank you so much. Any questions, comment from your side? I think it's
if possible. Yeah, I really wanted to, I was very interested in seeing the high number of patients who were on placebo that developed a de novo allosensitization. And I was just wondering if you have, if you can comment on why you think that occurred.
I think it's very difficult to understand. I don't know. We're surprised on the data. And I think we will finish in the next 3-4 weeks the correlation with the real clinical outcome on the individual patient, not only on the group, on the individual patient, to see is it just by chance or is it any relation to this? Because take in mind that for some markers we have not too many samples. It's still sufficient for making statistics, but of course it's not the complete things. In contrast to the allo, we have almost all, and for the cytokine we have also almost all patients analyzed as we had the sample, because it could be centrally stored and measured in a batch. And yeah, I think we also see is a relation between inflammation and allo response, is a relation between NOT cell and inflammation and T cell response inflammation. You have seen maybe on one slide that there's separation in 2 groups in the circulating NOT cells. One part completely normal after treatment, and one part a little bit better, but not completely normal. And it would be, of course, interesting. What does it mean that in some patients it go down? Because the method is relatively robust, and therefore I hope it's not a methodic issue.
Dieter, one comment for consideration. We should remember that the placebo includes albumin and DMSO, and probably what we see in the placebo is the effect of those ingredients. So it's not real placebo.
I agree completely, Sam. I think nobody knows exactly what does it mean if you inject, I think 30 or something like this, injecting with DMSO. Probably is not nothing. Something happens, and the question is what. And therefore we cannot, but that is a problem of the study. You have to do it because you have DMSO also in the cells, and therefore I think it's a proper control. Even maybe it's not the same if you would do nothing. I really surprised I was that the NOT cell response was so good in the PX cells, but not in the placebo, and so-called placebo, DMSO. But it's not so reflected with the clinical response. Meaning, I think it's important to learn, maybe endothelial cells alone is, or the repair of endothelial cells is not the complete story of getting clinical success. And at least something has happened. But yeah, and here I think we will, but the problem is there's not too many patients. It would be interesting to show the responder, non-responder on the individual levels to separate it. But I hope Guido can do it in the next couple of weeks.
Dieter, I would actually bring up one point that we didn't have. Norbert discussed more repeated infusion of cells, but I think what we also very extensively discussed before was earlier treatment. That maybe what also the inflammation at baseline indicates that our patient cohort is probably beyond treatment, that maybe we should go one stage earlier rather for 3 or 4. That the patients are more responsive to the treatment at this stage, although it might be probably difficult to justify, that they still have the capacity to respond in their endogenous cell profile and tissue composition to have a benefit from the therapy, which maybe also might be indicated by the circulating endothelial cells EPC analysis, that if the repair reservoir is still present in the patient, that you get a more beneficial effect of the cell therapy.
Yeah, I absolutely agree. Because these patients are very late stage and difficult to modify anything. The other thing is that inflammation obviously is associated with outcome. That's confirmed in PACE trial, as it has been shown several times before. In the phase 2 trial, as when I remember correctly, treatment with PLX-PAD cells resulted in a decrease in CRP levels, which did not happen in this patient. Yeah, that's the interesting difference, and maybe a reason for unsuccessful or lacking clinical efficacy.
That's also one of the best markers in the COVID studies, the reduction in CRP and IL-6 and increase in IL-10. And it also depends on the time of treatment and especially on the age of the patient. So if a patient below 60 years respond well to the MSC therapy IV in COVID, especially when earlier treated, that they're not beyond treatment in a very late stage critical disease, which is again an indicator that the cells might need something to function, like a repair reservoir in the patient, and the patient cannot be too severely sick that he's beyond treatment and already like highly inflammatory. And these patients are probably the ones who then die and have to be amputated.
Yeah, but that's a call for biomarkers to select patients that might profit from treatment. If
stratification, yes.
Yeah, if they're not in a hyperinflammatory state.
Yes, yes, absolutely.
Okay. Other questions, comments? Then I would like to hand over to Jackie to make a sum up and outlook. Please.
Thank you very much, Dieter, and thank you, everyone. I think it is important to mention that this PACE study, it was probably one of the largest, or maybe the largest, clinically double-blind placebo-controlled studies that we've seen in the last few years in this critical limb ischemia, which is an extremely severe disorder. Given the size and the fact that it was a double-blind, it gives a lot of information for the scientific community about the disease and the way to treat these patients. I do want to mention that also this study gave a lot of information for Pluristem about the mechanism of action of the cells and targeted the population in baselines, which of the patients have the best chance to respond. And that's something that may serve us to be able to better define populations and indications that are good candidates for our cell therapy and regenerative medicine. I think as we see here in this very good presentations that there are some very important outcomes for the study. Obviously, the final clinical outcome is disappointing because it didn't meet the primary endpoint as we wanted, and which is not enough for this severe disorder. But what we're seeing today gives me the understanding that probably cell therapy, young cells, and regenerative medicine is a good candidate for targeting and treating a critical limb ischemia, which is still as presented here, and we know it is a very significant unmet need. There is a lot of information specifically to understand sample size if we like to see a successful clinical study. We had the unfortunate situation that specifically in the placebo group, we didn't have the desired or the modeled outcome. But that's something that is very important for any clinical study that companies like we or other companies will conduct in the future, and we learn quite a lot about it. I think that it's a good time for me and for Pluristem to thank all the people that participated in this very important study. We were active in over 50 clinical sites in Europe, in the US, and in Israel, which involve a lot of medical teams and PIs that did this important study, the patients and families, all our partners in the Horizon that did quite a remarkable work with a lot of logistics, efforts, and challenges. The steering committee that supervised all this study, of course, Pluristem employees that worked very hard on that. And I do want to say a special thanks to you, Dieter, for your leadership in leading the space study, which, as I said, I believe it's very important. And obviously for the Horizon and that gives the opportunity to conduct this important study, and hopefully that's going to give some hope and better opportunities to understand the CLI and the way to treat it. So thank you very much.
Okay, thank you, Jackie. And I also like to thank all the people from the consortium. We have done it also in the last retreat, but here again, and particularly also for Inactor for organizing this meeting today. And to the European Commission for funding and for also the opportunity to extend this for 1 year. Anyway, it was an interesting experience, and we are now looking forward to the HipGene study. Thank you so much, and thank you, Norbert, for for participating and organizing the clinical part. Yeah, then have a nice day, all.
Thank you.
Very much.
Thank you.